Yeah, it's A. Good morning. I hope that you are all feeling well and your families are doing well. I'm sorry to not be there in person this week. I know that we all signed up to do this face to face, and it's not that much fun to be interacting with PowerPoint and videos online, but I will be back next week and I apologize for my absence. And again, I hope everyone is doing well. Today, we're going to talk about protein structure and function. And I left off last lecture with this website, link to this website and video, What is a Protein? A Biologist Explains. And I was very intrigued by the banner on their website. So that would be these images of proteins, because as a biologist, I see these and I recognize these as proteins right away. And I wanted to share all the different types of proteins and the diverse structures that are available to you as students in an intro biology class, so that when you saw something like this on the web or in a textbook, you can start to really analyze what you're seeing. You're seeing a folded group of amino acids reacting with each other based on R groups to make these shapes and forms, and each one of these shapes and forms has a function. So let's have some fun looking at the structure and function of proteins today. Thank you for joining me. On Tuesday, we left off with the video from the conversation. It was, what is a protein? A biologist explains. A protein is a molecule composed of amino acids. And like the biologist talked about in the video, it's a string of amino acids, and that the amino acids, they fold and they generate 3D molecules. In the written portion of this webpage, he has a description of different kinds of proteins. For instance, there's a description of collagen. It's a protein that gives the structure to our skin and our bones and our teeth. And then he talks about how keratin is in our hair and nails. It's a protein in our hair and nails. And that hemoglobin is the protein that carries oxygen around in our body. For today, we're going to cover primary, secondary, tertiary, and quaternary structure of proteins, how each of these are developed and function. We'll also talk about how proteins are catalysts, how enzymes work, and how the temperature and pH conditions of an aqueous solution change optimal conditions for enzymes. So to begin, here are 4 images that show the structure that builds a protein. Shown here on the left is the primary structure. So this is a string of amino acids. And the secondary structure is shown as an alpha helix. So we get these alpha helix structures developed when the amino acids in this string of amino acids build hydrogen bonds with one another. The next level of structure are these tertiary structures that are a combination of alpha helixes and beta pleated sheets. And quaternary structure is when 2 different proteins come together, 2 or more different proteins come together, and they fit together based on their chemistry and their shape. So to review, in words, the primary structure is the unique sequence of amino acids that make up the protein. The secondary structure is created by hydrogen bonding between portions of the peptide bonded backbone. Secondary structures are distinctively shaped sections of proteins that are stabilized largely by hydrogen bonds between the oxygen in a carbonyl and a hydrogen from another amino group. This generates alpha helix and beta pleated sheet structures. There's the tertiary structure, and there are 5 types of interactions. We'll go through that next. And lastly, there's quaternary structure, and this is the interaction of more than one polypeptide. The structure of proteins is generally described as having 4 organizational levels, as we've gone through. The first of these is the primary structure, which is the number and sequence of amino acids in a protein's polypeptide chain or chains, beginning with the free amino group and maintained by the peptide bonds connecting each amino acid to the next. So that's the beads on a string is what we're talking about. Here is the primary structure of insulin. It's composed of 51 amino acids attached to one another like a string on the beads. Pay attention to the book in these figures I show when we show a string of amino acids or polypeptides. We generally use the N-terminal on the left and the C-terminal on the right. So we start with an N-terminal and end with the C-terminal on this end. So again, this is an example of the primary structure of a protein called insulin. Secondary structure can make a alpha helix, shown here, where you have hydrogen bonding between amino acids. Hydrogen bonding also generates what is called a beta pleated sheet. So this string of amino acids bends this way, and the hydrogen bonds are shown here with the red dots, and then your amino acids come back this way, and they zigzag back and forth like a train track, and the hydrogen bonds are shown across these amino acid beads, across the track. Tertiary structure is dependent on 5 chemical interactions between the amino acid R groups. So we have hydrogen bonding, hydrophobic interactions, van der Waal interactions, covalent bonding, and ionic bonding. These are the 5 different chemical interactions that occur between R groups, which is why we emphasize so much on Tuesday about the chemical structure of the R groups and how they interact in aqueous solution, because this is what controls the structure of a protein, is how the R group interacts with other R groups. I've shown here on the left, here is an example of this is a picture or a schematic of beads on a string or an amino acid, a string of amino acids. We have polar side chains in blue and non-polar side chains in green. When this folds in on itself, you have a hydrophobic core with the nonpolar side chains, and then the hydrogen bonds can be formed to the polar side chains on the outside of the molecule. So all of the polar side chains are on the out, so the water or the aqueous solution would be on the outside, and the hydrophobic, the part that doesn't like the water, would be tied up in the middle. Van der Waal interactions between side chains of amino acids are a little, they're weak bonds, and they're similar to hydrogen bonds. And I'll get into that in the next slide. But what I wanted to focus on here is this amino acid, the string of amino acids here is this purple line. And if we were to have an R group on this side of this hydrogen bond and an amino acid on this side, so these are amino acids that are in this string, and the R group from this amino acid is interacting with the amino acid on R group from this amino acid on this side, and they're making a hydrogen bond. Here it shows that the amino acid on this side of the string of beads and the amino acid on this side of the string of beads is creating both a hydrophobic interaction and a van der Waal interaction, so that these two sides of this side and this side are tied together. This is a disulfide bridge generated because this amino acid has an R group with a sulfur, and this amino acid has an R group with sulfur, and they build a disulfide bond. And here is an example of an ionic bond between this amino acid with this R group, the amino R group, and this amino acid on this side that has a negatively charged oxygen. Van der Waals forces are transient, weak electrical attractions of one atom to another. Van der Waals attractions exist because every atom has an electron cloud that can fluctuate, yielding this temporary electric dipole, which means that one side will be more positive and another side will be more negative. This transient dipole in one atom can induce a complementary dipole in another atom, provided that they're really close, which is what happens when R groups get close to each other in a protein, when the protein starts to fold in on itself. These are short-lived complementary dipole interactions, and if the distance is just right between these clouds of electrons, then you get a neutral, also with the negative and the positive interactions coming together, then you get two electrical neutral closed-shelled atoms, which is what this image is showing on the slide, two electrically neutral closed-shelled atoms. Very close. It gives this net attraction because the dipole, there's a negative dipole, and then the positive on this side is the positive of this atom is attracted to the negative on this atom, and they're very short-lived, and if you get enough of these, then you get the ability for the protein to be held together temporarily in places. And this is really good for the lock and key formation, which I'll talk about in the next slide. So a few slides ago, I showed this drawing of a long string of amino acids depicted by this purple squiggly line, and I talked about the hydrogen bond here, the ionic bond here, and a disulfide bond here. But right now we're focusing on both the hydrophobic interaction and the van der Waal interaction that's occurring here. Again, although individually weak, numerous neighbor interactions in such central cores, so they're talking about this being in the bend of the amino acid string, can contribute a significant stabilization in the structure. Van der Waals forces can play important roles in protein-protein recognition when complementary shapes are involved. This is the case in antibody antigen recognition, where lock and key fit of two molecules yields extensive van der Waals attractions. So it's again, it's the electron clouds around atoms developing these dipoles, and the electrical interactions cause them to be attracted to each other for short periods of time. Get my laser pointer back. So tertiary structure is based on 5 interactions. This figure is in your book as well as in this slide right here, where the green ribbon is the long string of amino acids in the protein. They have an example of a hydrogen bond between the side chain and a carboxyl oxygen shown here. There's a hydrogen bond between two side chains. There's the hydroxyl here and the amide here. And then you have the hydrophobic interaction and the van der Waal attraction here between two methyl groups. 4 methyl, 2 on this side, 2 on this side, so 4 total. You have the disulfide bond here and an ionic bond here between the ammonia and the oxygen, so a positive and a negatively charged R group. I wanted to emphasize that the strengths of these bonds are very different. So the disulfide bond is the strongest. It's 167 kilojoules per mole. The ionic bond is the next strongest, followed by the hydrogen bonds and the van der Waals and then the hydrophobic interactions. So you'll need, again, hydrogen bonds and van der Waals attractions are strong only when there are quite a lot of them. So we will end this portion of structure looking at the quaternary structure of hemoglobin. I just wanted to remind you that we started today's lecture looking at these cartoon schematics of what proteins look like. This is what a hemoglobin protein looks like, and that these are many different polypeptides interacting with each other, and that's it's the combination of the string of amino acids and how the R groups are interacting with each other in both in the primary, secondary, and tertiary structures that ultimately leads to how a quaternary structure is formed. Just quick little note that when you see these cartoon schematics, to realize that these are not stagnant molecules, they're not hard, they are constantly in motion and they're moving around. And again, if you think about van der Waal interactions alone, those are things that are dynamic and they're constantly changing. They're swapping around electrons and they're moving around. So when you see these cartoons, to really think about, be critical about how you think that the coils are bending and opening and closing, and how the sphere might be coming this way or going that way depending on what aqueous solution is it's in, and also what other polypeptides are around it, whether they're going to come together or they're going to repel each other. Here are 2 videos I would like you to watch that are super helpful about, without words, actual diagrams and images of how these proteins are being built and how these proteins look in aqueous solution. And after you watch the videos, there is a musical. After you've watched those videos, here is a musical composition written by Augusta Reed Thomas called Dance Foldings. If you follow the link, you can listen to a 13-minute song that she has written, that was inspired by what we've been talking about the last couple of days, how amino acids are produced, how amino acids interact with each other to come together and build polypeptides, and how polypeptides come together to form proteins. And if you really start to read these notes, you'll see a lot of the things that we've been talking about in class, that the beginning of the song shows what she would imagine it would sound like, or what would be inspired her to write this part of the song. Is like a chain of amino acids. She has words in here like colorful and spry and kaleidoscopic, a ballet of materials being braided. It's interesting, and it's something that came out within the last year, and it's a unique musical composition written about biology, and I wanted to share this with you. A colleague of mine at Lummi Natural Resources, Suzanne Gray, she's the outreach coordinator for Lummi Natural Resources. She sent me a computer program that I've been testing out this week called Fold It. And I will bring my laptop in next week and each of you can take a shot at it. I've only briefly worked with it, but you can see that you can play around with how the R groups from each amino acid interacts with each other, and you can pull them together and take them apart. I think that it'll be useful for us to look at this so that you can see how these molecules really have to fit together quite nicely in order for the protein to be built and for the protein to function, and how easy it could be for it to not to work out. And maybe how easy it is that these things come together so beautifully. Now that we've talked about protein structure, let's talk about protein function. And one of the main functions is proteins act as catalysts. So what's a catalyst? Let's start there. Here is a graph talking about how reactants become products in a chemical reaction. Your x-axis is the progress of the reaction, and your y-axis is the energy required to make this reaction move forward. You see here that your reactants are at this energy level, and that in order for them to become products, you have what is called activation energy, and it is a peak. So it requires energy for this to happen, for this reaction to happen. So an increase in energy has to be put in. The blue is the activation energy without the catalyst, and the red line is an activation energy with a catalyst. And with a catalyst, it takes much less energy to go from a reactants to products. And this is what catalysts do. They help reactions move forward, and they lessen the amount of activation energy needed for a reaction to progress forward. An enzyme acts by, let me rephrase that. A catalyst is something that will change the activation energy required to allow a reaction to proceed. There is a figure in your book that looks similar to the one I'm showing here on this slide, and it's showing reaction progress on your x-axis and your Gibbs free energy on your y-axis. You have reactants, A and BC, and you have products, A, B, and C, and you have what's called a Gibbs free energy of change, which is less than 0, which means that this reaction is exothermic, that it will occur spontaneously. So, what is this activation energy? Why would an energy-releasing reaction with a negative delta G need energy to proceed? Well, that is because when molecule A and molecule BC come together, they develop into what is called a transition state, which has higher free energy than when the reactants were not interacting with each other. And the higher the activation energy, the less likely the reaction will proceed to products. So you have to find something, a catalyst, that will lower this activation energy, and that's what proteins do. So to go over this graph where we know that reactants come in close contact with each other, they go into what is called a transition state where they have a higher value of free energy than when they were not put together, and that in order to overcome this activation energy, the requirement for this energy input, we have enzymes that come in and help the reaction proceed. So I have here Pac-Man coming in, to sort of help the reaction proceed, because if you watch the video, the amoeba sisters have a really good analogy of how enzymes are a lot like Pac-Man. They have a perfectly formed mouth to grab the little balls in the Pac-Man game. Also, they use lactase as a enzyme in the human body that breaks down lactose. So I've included on this image here what lactase looks like, and also what the chemical, the molecular structure of lactose is. So, how do enzymes work? We know that certain reactions have an activation energy. This is the energy required to start a reaction. Some reactions are endothermic, so their delta G is, or endothermic, so the delta G is less than 0, and so it requires energy for it to proceed. Some reactions are exothermic, so the delta G is less than 0, so it gives greater than 0. So delta G, if it's greater than 0, it gives off energy. We also know that there is a transition state. So to get the bonds into a state that allows them to break, the molecule must be contoured, deformed, or bent into an unstable state. And that is how enzymes work, is that they have here's a picture from the amoeba sisters. I have the link to the video down here. It shows this Pac-Man looking enzyme. It has an active site where molecules can come in contact, and they have this image here of what that looks like. Here's the enzyme, here's the substrate, and when the substrate reaches the enzyme's active site, it has an induced fit. So I keep talking about how these proteins aren't stagnant, that they have this lock and key mechanism where certain substrates fit into active sites perfectly. But when that happens, it's not as though the enzyme stays hard and rigid. It moves and modifies and clinches on to that substrate based on chemical bonds. And when it does that, it makes sure that that's how the activation state is. Then the free energy is reduced, because then whatever reaction needed to happen can now occur in that active site. I've shown here a link, the YouTube link to a Jeremy Lecornu. I don't know how to say his name, but he does a really good job of reviewing what I've talked about as far as the going from reactants to products, what activation energy is. He uses words like activation energy, endothermic, exothermic reactions, transition state. He also goes into how these interactions between substrate and enzyme occur in a series of processes. So he introduces glycolysis, which is what we'll do. We will cover glycolysis in a couple of weeks. So it's getting you primed to think about enzymes and how they work together to help metabolic processes occur within cells. Something that we need to talk about is that these reactions between enzyme and substrate are regulated. Many of these enzymes require molecules that are not part of their molecular structure. These are called cofactors. These could be metal ions such as iron that are required for the enzyme activity to occur. There are also coenzymes involved. These are also sometimes required for the enzyme to work properly. There's also competitive inhibition, and this is when the activation site of an enzyme is blocked so that a substrate cannot react with the enzyme. There's a cartoon by the amoeba sisters I've included on this slide where you have an upset substrate saying, hey, that's my enzyme. And the inhibitor is saying whatever, and I don't see your name on it. So this is one way that the metabolic process is regulated through competitive inhibition. The other kind of inhibition is allosteric activation. So you see here an enzyme, and it has its active site here, and because the allosteric site has been activated, the substrate doesn't fit quite well. However, a competitive inhibitor does indeed fit that space really well. Something else that we need to talk about is that substrate activity is controlled by pH and temperature. So an enzyme that might be found in your stomach would really enjoy warm temperatures and high pHs, and it needs that in order to work properly. And if enzymes aren't in the at the right pH or temperature, then they are denatured and they won't work correctly. And all of this goes back down to the basic chemistry of those amino acids and the R groups, and how those are what are ultimately the foundation of what controls both the structure and the function of these enzymes. I'd also like to remind you that on Tuesday and last Thursday, we talked about the origin of life and how scientists are working really hard to figure out where life originated, and that they have yet to be able to prove that proteins were the first signs of life. They can't do Miller-Urey experiments in the lab and produce protein from nothing, and that really they think that it's nucleic acids that are responsible to that. They are what created the template for the origin of life on Earth. And I thought that would be a nice little thing for you to think about, is that what was the first true organic compound that gave the template for life to begin on Earth? Was it a protein? Was it a nucleic acid? And maybe after next week, when we go over ribonucleic acid and deoxyribonucleic acids, you'll be able to better argue your points. Was it protein, or was it amino acids, or was it a nucleic acid that was first generated in those supposed deep sea hydrothermal event situations, or that began all of this, began the science of life, of biology. Thank you for joining me remotely this week. I can't wait to see you in class again, and let me know if you have any questions, and shoot me email, contact me on campus, and I will see you next week.